What if I told you the hardest part of developing a Leuprolide Depot isn't the peptide, it's controlling how plastic breaks down inside the human body for 3 months without destroying the drug. That's the real challenge behind long-acting injectable depots. Hi, I'm Raghuveer Singh Parmar, Director Scientific Affairs at ResolveMass Laboratories, Canada. As a CRO and CDMO focused on complex drug molecules, ResolveMass Laboratories delivers precise, science-driven solutions for peptides, impurities, and regulatory critical analytical challenges. When most people think about injectable drug development, they imagine a simple, sterile solution. Dissolve the API, filter, fill, and you're done. But depot formulation like Leuprolide Acetate are a completely different world. Here you are not just developing a drug, you are engineering a controlled release delivery system made of biodegradable polymers, microspheres, and a peptide that really doesn't want to stay stable. You are essentially designing a tiny drug factory inside the patient that releases medication slowly over weeks or even months, and that's where things start getting complicated. Let's say you are developing a 3-month Leuprolide depot. Everything looks good in early formulation screening. Then you run your first in-vitro release testing, and suddenly 25% of drug releases on day 1. That's the burst release, and now your entire pharmacokinetic profile is off. So what happened? Maybe the polymer molecular weight shifted slightly, maybe the solvent evaporation rate changed, maybe microsphere porosity increased. Tiny changes, massive clinical impact. This is why depot formulation development is really a microsphere engineering problem, not just a formulation problem. To solve this, you need multidisciplinary expertise: peptide chemistry, polymer degradation kinetics, encapsulation science, particle engineering, release modeling, analytical characterization. If even one of these pieces is missing, the entire system becomes unpredictable. And unlike oral generics, you can't just match dissolution, you must match particle morphology, polymer composition, release kinetics, peptide stability, IV-IVC correlation, and that's very high bar. Real world formulation pain points. So in real development programs, we usually see 4 major technical issues. First, burst release from porous microsphere, that's the first challenge. Second, peptide degradation during encapsulation, because peptide is easily unstable and easily degrade. Third, particle size distribution variability, and the fourth one is scale-up inconsistency. And interestingly, scale-up is often where everything breaks. A formulation that works perfectly at 10 ml suddenly behaves completely different at 10 liters. Why? Because mixing energy changes, emulsion droplet size changes, solvent removal rate changes, polymer precipitation changes. And now your microspheres are different, where analytical expertise become critical in this scenario, because this is actually where the strong analytical capability becomes just as important as formulation science. At ResolveMass, we have seen cases where 2 depot formulations looked identical visually, same particle size, same loading, but release profile was completely different. The difference? Internal microsphere morphology. Using SAM imaging and detailed release profiling, we identified subtle porosity differences caused by solvent diffusion rates. Without that level of characterization, the root cause would have been missed entirely. And this is why analytical driven formulation development is essential for complex depots. Let's talk about what actually controls release kinetics. Three key formulation parameters: polymer selection, drug loading, and microsphere morphology. Polymer ratio, PLGA 50-50 versus 75-25. This changes degradation speed. Molecular weight changes release duration. Drug loading affects diffusion pathways. Particle size controls surface area, and porosity controls water ingress. You are basically tuning multiple knobs simultaneously, and they all interact with each other. For example, increasing drug loading may increase burst release. Reducing particle size may increase degradation rate. Changing solvent may alter peptide stability. This is why formulation design becomes an optimization exercise. Another underestimated factor is microsphere manufacturing technology. Solvent evaporation is widely used but sensitive to mixing conditions. Phase separation improves loading but can affect morphology. Spray drying is scalable but may stress peptides. Microfluidics offers precision but scaling is challenging. There is no universal solution. Technology selection depends on peptide sensitivity, target release duration, polymer type, scale requirements, scale up reality check. Here is something rarely discussed. Scaling a depot formulation isn't linear. You cannot just multiply batches. You must redesign mixing strategy, solvent removal, drying process, filtering approach, fill finish conditions. Otherwise, your commercial batch will not match your clinical batch. This is one of the biggest risks in depot development. So let's talk about analytical characterization. So regulators now expect deep characterization for depot generics. Particle size distribution, drug loading uniformity, peptide stability, residual solvents, in-vitro release profile, IVIVC modeling. We have worked on programs where release testing alone ran for 90 days per batch. That means analytical strategy directly impacts development timelines. A poorly designed release methods can delay an entire submission. That's why early analytical planning is critical, not something to add later on. This is where integrated CRO CDMO partnerships becomes powerful and really valuable at this stage. Instead of formulation team in one place, analytical team somewhere else, manufacturing at a third site, you have a coordinated development flow. Formulation decisions consider scale up, analytical method support release modeling, process development aligns with GMP production. This reduces technology transfer risk, batch variability, timeline delays, and most importantly, regulatory surprises. So overall, this reduces a lot of time and efforts from the team of scientists considering the development and analytical and formulation development. So if you are developing a Leuprolide Depot or similar peptide injectable, the most important things to remember are depot formulations are delivery system, not just formulations. Microsphere morphology drives release kinetics. Scale up changes particle characteristics. Analytical characterization is critical for success. Integrated CRO and CDMO support reduces development risk. If you are navigating complex drug development challenges from IND enabling studies to NDA submissions, connect with ResolveMass Laboratories to accelerate your program with confidence. Thank you so much for watching, and see you in the next one.